part 3, we're going to talk about DNA replication. And just as a side note, my son is here with me, so you may get a little bit loud as he likes to scream or throw things. Alright, anyways, learning targets we're going to hit today. Understand the base pairing rules, understand base pairing and its role in replication. So that's what we're really going to focus on. Transcription, translation, that comes later. 6, understand that replication is semi-conservative, so that's a vocab word you're going to need to know, and occurs on the leading and lagging strand. And you're going to need to know the role of enzymes in replication, helicase, primase, DNA polymerase, and like. Okay, so first we're going to talk about DNA replication. DNA in eukaryotes is found only in the nucleus. When DNA is replicated, it does not begin at the beginning and work its way all the way through the DNA molecule to the end. Okay, DNA molecules are really wrong. Replication occurs at several origins. Origins means like beginnings or spots. So there are several spots where replication begins, and it happens at multiple places along the DNA chromosome. And we'll talk a little more about that when we're talking about gene control. Alright, DNA replication, it occurs during the S phase of the cell cycle. So if you remember our two units ago, when we were talking about cell cycle, we said that S stands for synthesis. That is where DNA is being synthesized or it's being replicated. And when DNA is semi-conservative, which means when DNA is replicated, we have the original strand here, okay? The original strand here, and semi-conservative means we're going to have one old strand, okay, and then we're going to have one new strand, okay, one old, one new. This is going to then be our new strand of DNA. Okay, not too complicated. Semi-conservative, it's semi-new. Okay, semi-old, semi-new. DNA replication enzymes, there's a lot of these that you need to know. I suggest maybe doing flashcards. DNA helicase, that's the first one we're going to talk about. If you take a look at the picture over to the right, I'm going to circle right here. DNA helicase, it's an enzyme that basically unwinds and then breaks the hydrogen bonds between the nitrogen bases. Okay, unwinds it, breaks it, it creates what's called a replication fork. So if you see here, replication fork, this is when the two strands of DNA are going to be separated, like if you were looking at a fork in the road. Okay, there's my really bad impression of a fork. The unzipped bases, or the bases that have had the hydrogen bonds broken between them, okay, so a base like this that's been unzipped, is going to form new hydrogen bonds with free nucleotides that are floating around in the nucleus. Okay, then primase is going to assemble the RNA primer. This picture doesn't show up, the next one I'll point it out for you. Okay, this is really important because this is what allows DNA polymerase, and DNA polymerase is like our glue factory. It's what's going to come in and glue or put together, cement factory probably be a better analogy there. It's going to come in and cement or bond the sugar to the phosphate, because if you remember when we're talking about structure, we have this alternating phosphate deoxyribose sugar, phosphate deoxyribose sugar. So these bonds here that bond them, those are covalent bonds. They're strong bonds. That's why it's called backbone. And DNA polymerase is what comes in and bonds those sugars to the phosphates, connecting them. Okay, and then there's DNA ligase, which we'll talk about when we talk about Okazaki fragments. Alright, accuracy. It's extremely important for this process to be accurate. Because if you remember when we did the activity with the noodles where we stuck the pins in there, the push pins, and then we folded them, we had folding rules, and depending on what shape your structure was, that was what protein you had. Okay, so the sequence of amino acids is what was different with each of those, because each of you put in your own sequence. Some of you like to put all the reds and then all the yellows. Some of you like to mix them up. Okay, that sequence of amino acids, that comes from the sequence of DNA, and we will talk about how we get from DNA to amino acids when we get to translation, which is part 5. Okay, so this process is very accurate. There's only about 1 error in every billion nucleotides. That's incredible. Okay, and the reason that it could be so accurate is that there are 20 repair enzymes that go in and they edit goofs. Or if you have something like here, that's not correct, because we know that adenine is always going to be bonding with thymine. So adenine cytosine, it doesn't work. It's going to give us this bow here. Okay, and so what we're going to have happen is we're going to have one of these repair enzymes come along and go, you do not belong here, kick it out, fix it, put a T in there. What causes these mistakes? Well, there's several reasons. Sometimes mistakes just occur, random mutations. You have mutagens, there are things like heat, excessive heat, like when you go get sunburned. The reason that can cause skin cancer is because you're going in and you're damaging your DNA here. Chemicals, radiation, those are all mutagens that can cause mistakes to occur more frequently, and to occur. So exact sequence is very, very, very important, because that is what makes you who you are. Makes your DNA unique. Okay, Okazaki fragments. So what is this? Well, in order to talk about this, we need to discuss what anti-parallel means. You all should know that parallel means something like this. 2 lines never touch, right? Okay, it is. Anti means opposite. So anti-parallel means the 2 strands of DNA run in opposite directions, and then we know that they're bonded together by there's nitrogen bases in between. Okay, so the sugar phosphate groups point, quote unquote, in different directions. We have the 5' to the 3' strand, that's your leading strand. Then we have the 3' to the 5' strand. Okay, now before I get any further into this, I told you guys I would point out the primase. There's the primase in this one, and it's what's going to allow the Okazaki so it's going to allow the DNA polymerase to come in and get started. Okay, so normally DNA polymerase assembles a new strand from the 5' to the 3' end. So if you see here, here's my new strand, and it's going from 5' to 3' end. Okay, and that's its normal direction. DNA polymerase, while it is amazing, cannot go backwards. So the leading strand is okay because it can build from the 5' to the 3'. It can build the way that it needs to build. That's normal. Okay, Okazaki's fragments are normal too, but the lagging strand is going to form in these fragments because the lagging strand wants to go from the 5' end to the 3' end. Okay, but it can't do that. It wants to build, in the 5' to the 3' end, but it can't do that because it's going to have to start. It can't start over here. It's got to start where it's being broken here. So what's going to happen is this DNA polymerase, is going to come on and it's going to go and build these little fragments, these little Okazaki fragments in here that will then be tied together into one complete strand by DNA ligase. Okay? That's it. See you next week. Or not next week, next section.